In this video, we continue our exploration of quorum sensing, the process by which bacteria, as they go from low-density environments to higher-density environments, accumulate molecules known as autoinducers, such as, for example, the acyl homoserine lactones. We're at a low density, where we have just a few of these pink bacterial cells. There's a correspondingly low density of the red diamond shapes, which are the autoinducers. As the cell density increases, the concentration of those autoinducers increases as well, ultimately resulting in the change in behavior, such as the formation of a biofilm, a thick mucus-covered layer of bacteria where the bacteria are enshrouded in an extracellular matrix that promotes their survival in challenging environments. So we are going to take a closer look at the molecular basis for this process here, evaluating how this takes place. How is it that a high density of cells and hence high density of these acyl homoserine lactones triggers changes in the behavior of the bacteria and changes the bacteria to behave in a more coordinated group manner. So looking at this diagram, as we evaluate what's going on in the cell, in the previous video, we referred to the acyl homoserine lactone synthase as our LuxI. That was the enzyme that catalyzes the formation of the acyl homoserine lactone from S-adenosylmethionine and the thioester that is tethered to the acyl carrier protein. And so what happens within a cell, as we look more closely at this phenomenon of how quorum sensing is happening at the molecular level, is that the enzyme LuxI takes the precursors, which were our acyl carrier protein tethered structure, plus the S-adenosylmethionine to form the autoinducers, the acyl homoserine lactones, which I'm going to abbreviate as AHLs. Those exit from the cell, where the cell is pink, and escape into the surroundings, into the environment around the cell. Then, when acyl homoserine lactones come into contact with additional cells, they enter those cells via diffusion through the cell membrane. Once they are inside the cell, that is where we are going to see the biological effect triggered. Once the acyl homoserine lactone enters the receiving cell, the acyl homoserine lactone that we see here as the red diamond schematically is going to bind to a particular protein that in the case of the acyl homoserine lactones is referred to as LuxR. You could think of the R here and help you remember it as this is the receptor. It is going to be receiving the acyl homoserine lactone molecule. What happens is that once LuxR has interacted with and bound with that acyl homoserine lactone molecule, that is going to change the conformation of LuxR, the three-dimensional shape of LuxR, in other words. That will enable LuxR then to bind to the Lux promoter region that is immediately upstream of genes that are under control of this regulator, LuxR, and thereby trigger the expression of those genes. So that's what we mean when we say that the LuxR complexed with the acyl homoserine lactone is binding to DNA, is that the LuxR that we have here in complex with our acyl homoserine lactone, so we'll call this LuxR that is bound to the acyl homoserine lactone, that together is going to result in a conformation change of LuxR, enabling LuxR in complex with the acyl homoserine lactone to bind to the Lux promoter. And we can represent this schematically to illustrate what I'm referring to here by thinking of this black line as being the DNA of the organism. plug in here in yellow a gene that is going to be under the control of the Lux regulator. And so upstream from that gene is the Lux promoter. And what will happen is that the LuxR, this blue protein here, will bind to that Lux promoter only when it has the acyl homoserine lactone there associated with it. So that acyl homoserine lactone in red changes the conformation of LuxR, causing LuxR to bind to that DNA of the Lux promoter that is immediately upstream of the gene that is under control of LuxR. And then at that point, what happens is that once LuxR, which is bound to the acyl homoserine lactone, binds the promoter, that gene is expressed. And that gene is involved in things like biofilm formation, virulence, conjugation, and the wide variety of other different traits that are associated with quorum sensing. And so the LuxR plus the AHL binds to a variety of Lux promoters that are upstream from the genes that they control the expression of, so that there are a variety of phenotype changes that occur, phenotype meaning visible changes such as the production of light or the formation of biofilm, that we can observe in response to the acyl homoserine lactone concentration increasing in these. On the other hand, if we have LuxR that is not associated with the acyl homoserine lactone, that LuxR, which I'm drawing out here, does not interact with the Lux promoter because it does not have the proper conformation. So it's essential that the acyl homoserine lactone come in and bind to LuxR in order to activate the genes that are under the control of the Lux promoter here. And so in that regard, the genes that are under this control are the genes that are involved in the coordinated group behaviors. So that once the concentration of the AHL goes up to a sufficient level, we will see the changes in gene expression that result in the formation of biofilms, the production of light, and the wide variety of other traits that we associate with quorum sensing. And as this process continues and the cells continue to grow, divide, and multiply, the behavior can become intensified because these cells continue to use LuxI, the biosynthetic enzyme, to generate additional acyl homoserine lactones. Those exit the cell, and then the process repeats. So as the concentration of cells continues to increase, the amount of acyl homoserine lactones continues to increase as well, and the extent to which traits are ultimately impacted will continue to grow also. So now that we have looked at how the acyl homoserine lactones yield the observed effect on the phenotype of the organism, we're going to continue in the next video to explore the possibility of inhibiting quorum sensing as a promising avenue for drug discovery.